All right. So today we'll be finishing our part on LFP analysis, and then we will start with EEG. So I'm assuming that everybody got the assignment. Uh, did you guys all see the assignment?
Yes, sir.
Yes. Okay. Yeah. So you've got now 2 weeks to do that assignment. It's a MATLAB heavy assignment. So please get going soon. All right. So we have done all of this. We did coherence analysis, and now we are going to again, we are going to quickly do the part where we were going to look at the relationship between spikes and LFP. Are there any questions on coherence analysis that we did in the last class? Okay, so if everyone is happy with that, let me just um, very easy way to look at whether the relationship between spikes and look is to compute triggered average, which is simply the average LFP around a spike. That's it. So if you've got a signal that LFP signal that looks like this, and suppose there's an oscillatory component and spikes happen to occur at a particular phase, let's say at the trough of the LFP. So if you take these segments around a spike, this segment here, this segment here, that segment there, and just look at the average of that, you will see a dip like this. But if they're occurring at random phases around the LFP, then you won't see anything. So that's the idea. If you look at the spikes, spike triggered average during the for the spikes in the baseline period before stimulus onset, you get a trace like this green trace here. This is baseline. And during the stimulus period, you get a trace like this, and you can maybe vaguely see that there's an oscillation like that here, which means that spikes tend to be occurring at a particular phase of the gamma wave. Okay, and there's also this sharp dip here at time 0. 0 is the onset of spike by definition, because you're looking at spike segments around, LFP segments around the spike. This is nothing but, so all the events that led to the spike will trivially be captured here in the LFP. So any synaptic event as well as a spike bleed through. This term keeps coming, spike bleed through. So what does this mean? Think about what is happening in the LFP. So LFP is recording um, all the the potential generated because of transmembrane currents. So some synaptic activity would have led to depolarization of the neuron, which led to a spike, right? Which you record in the extracellular potential. Those synaptic events which led to the spike are always locked to the spike quite trivially, because those synaptic events led to the spike, right? So those would have some sort of a you know some sort of a signature in the LFP. So that's synaptic activity that led to the spike. In addition, when there is a spike, remember the intracellular potential is like this. It's a very sharp sort of a transient, 1 millisecond or so, but it's like a Gaussian. It's not a zero mean signal per se. It's like a Gaussian. So in the frequency domain, it will have power at many frequencies, including low frequencies. Okay, so the low frequency part of the action potential is called spike bleed through. It's essentially what you record in the extracellular potential at lower frequencies because of the low frequency components of the spike itself. In the LFP, you will see some as if the high frequency spike bled through in the LFP, and that is what. So this is both synaptic activity and spike bleed through. And in extracellular potential, there's no way you can sort of dissociate between the two. If you do some modeling that some people have done, they say that the contribution is about half, which means synaptic as contribution of the LFP about contribute half to have equal contribution in the LFP. But that's a point. The main point is that the LFP signal is to some extent corrupted by the spike, which you can see here as a sharp dip at time 0. Okay, where 0 is the onset of the spike. So if you look at spikes and LFPs from the same electrode, you see this bleed through, and then there is this oscillation as well. Same for the spike and LFP from the second electrode. But if you look at spikes from one electrode and LFP from the other, then you don't get this bleed through because spikes are not occurring exactly uh, um, when one electrode is picking up spiking activity, the neighbor is not picking up that spiking activity, right? Because the spikes activity is more sort of local to one electrode. Unlike the LFP, which seems to have high coherence across electrodes, so this oscillatory activity can be picked up. So often a good strategy, therefore, to pick up just the oscillatory contribution is to look at spikes from one electrode and LFP from the other, which is what is shown here. And then you can see some part. Of course, there's some bleed through, but it's much less than what you have from the same electrode. Okay, and now you can compute the Fourier transform of the LFP, FFT of the ST LFP, spike triggered LFP. And if it shows some peak at 40 hertz, let's say, then you would say that spikes are occurring at a particular phase of the LFP at 40 hertz. Okay. Any questions on this? Is this fine?
Sir, could you repeat the bleed-through concept again? I couldn't
Yeah,
understand.
bleed through concept simply means that when so here's a neuron and you've got an electrode close to it, and when this neuron gets some synaptic activity, some neurotransmitter release, it's going to open some ion channels and some current is going to come in, and that's all synaptic. But if it reaches a threshold, it generates its own action potential, right? So we had done that. Now that action potential, when it generates, so intracellular, this is how it looks like. This is time and this is voltage Vm, goes from -60 to +50 or whatever, right? And this thing will also have an extracellular potential outside the electrode because there are some currents that are flowing in and out to generate this action potential. And this structure or this event has power at many frequencies, not just at higher frequencies. There is some contribution even at lower frequencies. Okay, and that even when you filter the signal in the lower frequencies to get the will have some contribution. That is called spike bleed through. This is just the low frequency component of the spike itself. That's it. So it's going to have this little dip here, which is localized to the onset of the spike. And in the frequency domain, this thing will have, uh, this thing will have uh, um, uh, presence in the LFP because it's like a very sharp Gaussian. You can actually model it as a LFP oscillation plus a very sharp Gaussian. And this thing in the frequency domain is also a Gaussian like this. If it becomes a delta function, it becomes flat here. But if it's a Gaussian, it is a Gaussian in the frequency domain whose sigmas are sort of reciprocals of each other. Which means even at low frequencies, 0 to 200 hertz, which is your LFP range, 250 hertz, you will see some effect of the spike itself. This is spike bleed through. And if you can reduce this, then you will actually get a bigger contribution from the LFP, the oscillating part of the LFP. So that you can do by recording LFP from one electrode and spikes from another electrode, and then looking at the ST-LFP. It's a common strategy. It has its own flaws also, but it does reduce this sharp transient that you see when you have spikes and LFP from the same electrode. Okay.
Okay.
So in general, instead of just using the Fourier transform of the ST-LFP, we do something comparable or something similar, which is called spike field coherence. One reason is that it puts it, in the same sort of, architecture, some sort of same similar code as the field coherence that we had done earlier. Field coherence. So remember, what was field coherence? You had LFP signal from electrode 1 and LFP from another electrode, and you were looking at the cross spectrum. So if this signal you decompose it into a bunch of oscillations, and each one is some oscillation like this, J phi F, and this is B, BF J theta F. You would take the cross spectrum. So the cross spectrum would be AFBF and then e to the power j phi minus theta. So it's a vector whose amplitude is the product of the individual amplitudes and a phase, which is the difference of this. And then you average it across trials. So if the phase are aligned, then you get a large component. If they are not aligned, they're all over the place, you get a component. And then you divide by the auto spectra, which will normalize the AFs and the BFs. That is what you did for field coherence. In spike field coherence, all you do, because you don't have two LFP signals, so suppose you have got spikes from electrode 1 and LFP from electrode 2, the spike signal is just thought to be like an LFP signal with a 1 wherever there is a spike and 0 elsewhere. Okay, so consider the spike train as an analog signal with ones at positions when there is a spikes and 0 elsewhere, sampled at the same rate as the LFP. And then you consider this as an analog signal and compute the coherence between LFP and the analog spike train. That's it. No magic here. So the spike train, you convert spike times to bins, which means you convert it to a time series with ones and zeros, and then you compute the coherency between the signal and the uh, analog spike train, and that gives you what is called the spike field coherence. So why does this work? Let's just think about this a little bit. I guess I can just draw it, make it here only. I'm going to open a Jamboard, but let me just do it here. So the idea here is very straightforward. Uh, so see, when you are multiplying the spectra, so you are multiplying xf, y star f, right? That means in the time domain, you are convolving the two time series. There's a conjugate here, but essentially it's a convolution. Now, when you have a spike train, which is a bunch of delta functions, and you convolve it with an LFP signal, essentially for each of these spike trains, each of these spikes, it's just this plus that, and so on. Convolution, because of shifting theorem, will just give you a segment of the LFP centered on the spike. And you do that for each of these spikes, and this spike train is just the sum of those. So it is in some ways doing a computation similar to the STLFP, right? Because essentially you have converted your spike train to an analog signal. So here are your spikes and here's your LFP, and multiplying the cross spectra of these is uh, multiplying the spectra of these equivalent to taking the convolution of this with that, and that convolution is actually going to be comparable to taking the STLFP, because convolution with a delta function by shifting theorem essentially just takes the spike segment around that LFP segment around that spike. So this is just a fancy way of taking the Fourier transform of, but then you also normalize it some way that normalizes the energy of the LFP as well as the number of spikes that you have. So your spike field coherence is just in principle very similar to taking the STLFP and then doing some sort of normalization to bring the values between 0 and 1. Okay. So in your data, the data that I've given you, if you do the STLFP, you compute the STLFP for the baseline period and the stimulus period and you plot them, you will get plots like these. This is with a single taper and this is very noisy. So spike field coherence in general from a pair of electrodes is always very noisy. Unlike your field coherence that looked sort of nice here, this was field coherence. Spike field coherence looks very ugly when you record from a single electrode. But one thing that you can see that when you have spikes and LFP from the same electrode, it sort of tends to increase as frequency goes up, that you see here. And when you look it from a neighboring electrode, it sort of falls down. It's not really happening here very well. But in general, this increase is because of this guy here. This thing here. When the you compute the Fourier transform of this, it will have high energy in the at higher frequencies, which are trivially locked to the spike. So that is what produces this sort of a response. And there is a peak at 50 hertz, but then there are also a lot of other. So for a single pair of electrodes, it's hard to make sense of this. Now, unfortunately, in your case, this is all the data that you, but I had data from lots of other electrode pairs. So if you average all of that, no, this is not it. This is just the SFC with 5 tapers. When you have 5 tapers, then you sort of see some increase in the gamma range in all cases. And when you have LFP and spikes from the same one, there's also an increase at higher frequencies, which you don't see in this. But now if you average a lot of spike LFP pairs from different electrodes, you will end up a plot like this. End up with a plot like this. And this clearly shows that spikes are locked to the LFP at gamma frequencies. These are three different contrasts. You have data only for the 100% contrast. So my plot here, this red plot, is actually the average of many red plots um, like these. Okay, if you average them, you will end up with a plot like this. And this is some 130 or 150 electrodes. These are for two different monkeys. And if you look at the spike LFP phase histogram, which is similar to what we have done here, the LFP phase histogram, the same as this now. Spikes tend to be occurring at a particular phase, which is slightly off 180 degrees. And in fact, if you look at the ST LFPs, you see that they tend to occur just 1 or 2 milliseconds before the trough of the gamma wave. There is a gamma wave. You can see the gamma wave here. Spikes are occurring close to the trough of the gamma wave, which is represented as a histogram which is slightly shifted from 180 degrees. And this histogram is for this particular frequency at the peak of it. Okay, so there is a peak, and the peak, when you look at the histogram of phase distribution around that peak, you get a histogram like this, and this is the STLFP. They are all essentially saying the same thing, that spikes are weakly locked to the gamma rhythm. I say weakly because the coherence value is rather low. It's a big, small increase, but nonetheless a significant one. Okay, any questions on this? This is spike field coherence analysis. Any paper you pick up which has spikes and LFPs, you would see this analysis being done. And it just tells you whether spikes are coming at a preferentially at some phase of the LFP. And if so, the phase consistency will be shown by this bump. And then you can look at the phase distribution around at a particular frequency, and it will not be uniform. A uniform would be a circle like this. Non-uniform would mean that it's coming at a particular phase, so it's non-uniform like that. And you can get a sense of this by looking at the spike triggered LFP also, as shown here. Okay, and getting this plot using Cronux is quite trivial. You just compute the spike, convert the spike train into an analog data, and then essentially compute the field feed average, where the field is now a proxy of this, spike converted to a field. Okay, and if you had recorded here, I have the luxury of having multiple electrodes recorded at the same time, so that I can take spikes from one electrode and LFP from the other. If I did not have that luxury and I was only recording from one electrode, this plot typically would have looked like this. So from 0 to 80, you will see the peak, but later on it would just increase like that. And this is that contribution of the spike triggered, spike bleed through that you see here. This is what you typically see when you do spikes and LFP from the same electrode. When you do it from different electrodes, it flattens, it becomes low. And this thing goes away. Okay, folks. So now, if you think about what I have told you so far in the real data, I think there's a reason to be a little bit puzzled. And the puzzling part is this, that what I've given you two data from two electrodes, spike 1, LFP1, spike 2, LFP2. So there's spike train 1, spike train 2, LFP. I'm just showing you the 40 hertz component, let's say. So I have already shown that in the LFP, there is a strong gamma rhythm, and the gamma rhythms are linked in the sense that they are occurring at a particular phase of each other. We saw that here, occurring at a particular phase. Now, this data here shows that spikes are occurring at a particular phase of their respective LFPs. In fact, spikes are in the respective phase of a neighboring, which means that the spikes must be coming at a particular phase of each other, both of these. This spike train is occurring at a particular phase of this LFP, and it's related to that LFP, and spikes are occurring at these spikes are also occurring at a particular phase. So that means these two spike trains should have some phase relationship with each other as well. They must be coming in a particular sort of time relative to each other, because they are both related to the LFPs that we have recorded from the two electrodes in a particular way, and these two LFPs are sort of phase locked. But how come when we did the analysis in the first module, everything was Poisson-like? Remember, I told you that it looks all Poisson-like, but in the next module, I will show that they have a particular relationship with each other. This is that part. So I have shown you that the signal, spikes. There is in the LFP a very strong gamma signal. And now I am showing you that the spikes are not blind to that gamma signal. They are actually occurring at a particular phase of the gamma. So it's not that they are firing in a Poisson-like manner. So how do we explain this discrepancy? How come in the first module we thought they were all Poisson-like? And now I am saying that they are not Poisson-like. So the answer lies in the technique that we were using. So in the next two slides, I will just quickly explain what this is, and that will be the end of the spike LFP part. So here's a simulated LFP. This green is a simulated LFP, a simulated spike train and LFPs. So here spikes are always occurring at the trough of the LFP. And then there are some spontaneous spikes as well, but very few. Okay. And these 10 traces correspond to 10 different trials. So now, if I look at the spike triggered average, which is the average LFP locked to the spike, that's the green trace here. Of course, you'll get a nice sinusoid, which correspond to this oscillation. Remember, the time scale is different. This is minus 50 to 50. This is 0 to 200. So the spike triggered average catches this oscillation. The autocorrelation of the spike trains, because they are occurring periodically, has this nice oscillatory structure. So there are peaks coming at 0 and also at 25 and 50 milliseconds. And the spike spectrum also shows this. So you have got a nice peak at, in this case, at 20 hertz. So everything is good. The latter two, the autocorrelation and spike spectrum, are computed just from the spike trains, nothing else. The top two, the spike triggered average and the spike LFP coherence, are looking at relationship between spikes and LFPs, and everything shows you the same thing, that spikes are locked to the LFP. So you've got, this is that green trace, hard to see. So it's nicely showing that spikes are occurring at a particular phase of the LFP and locked to about 20 hertz, or maybe I think it's 25 hertz. Okay, so either if you had done the spike field coherence analysis like this, or just the spike analysis like that, you would have got the same result. But now consider a situation where spikes are occurring at the trough of the LFP, but with some jitter. See, there is jitter here. Still at the trough, near the trough, but with a little bit of a jitter. How does that affect the measures that I am looking here? Now, if you think about the spike triggered LFP average, I take the spike and look at the average. It's still more or less at the trough. It's only a 2-3 millisecond shift here and there. So it's always at the trough. So the spike triggered average of the LFP is still more or less sinusoidal. The amplitude has decreased a little bit, but it's more or less similar. So the spike field coherence is also more or less similar. You can think of the spike field coherence as approximately the Fourier transform of the spike triggered average. So as far as the spike LFP coherence is concerned, you are still catching this relationship. But what happens to the autocorrelation? Here now there is no LFP. I am just looking at the autocorrelation of the spike train. And in this case, a little bit of a jitter causes a big drop in this peak here. Okay, this jitter is essentially, spreading out this very sharp peak to a very sort of broadish peak. So that now when you compute the spike spectrum, there's actually quite a lot of spike spectrum is just the Fourier transform of this. There's quite a lot of reduction. Still there, but it's reduced. Now consider a case where it's occurring at the trough, but not in each cycle. It's occurring once in like 3 or 4 cycles of the LFP. Whenever the spike occurs, it occurs at the trough, but it's not occurring at each trough. It's occurring at some troughs of the LFP, not all. How does that affect these different measures? Now, for any spike LFP measure, okay, so I am looking at what is happening to the spike near the LFP. Because all spikes are occurring near the trough, the spike triggered average actually still captures it. See that? Still there. Because whenever you get a spike, it's occurring at the trough of the LFP. It's a different matter that it's not occurring at each trough of the LFP, but whenever it occurs at the trough. So the spike triggered LFP average catches that. So the spike field coherence still catches that. Still there. However, if you look at the autocorrelation, because it's not coming rhythmically at each cycle of the LFP, if I've got a, look at one trace, it's not here, it's just coming here after the, like after the third cycle or something. and in a different trial, it will come at a different one. The autocorrelation function looks terrible now. It has lost this rhythmicity. So if you just looking at the spike spectrum of the autocorrelation, it has now become flat. Just by looking at the spikes, you can't tell whether there is some rhythmicity here. This is what we did in the first module. We were just looking at the spikes without this clocking, signal which is present in the LFP. And without that, it all seems random. Autocorrelation is flat, cross correlation is 0. You look at this and say, hey, it's all Poisson-like. But if you had access to the LFP, which is this common sort of rhythmic cycle present, you would see that spikes are occurring occasionally to the trough of the LFP. There are also lots of spikes that are not occurring at the trough, but some spikes do occur, which can be captured in the spike field coherence, as done here. Okay. So the spike relationship, sorry, whether there is rhythmicity or not, you have to work with spike field coherence. Spike coherence gives a peak only in rare cases, when the spike coupling is very strong and spikes are occurring at each or almost all the troughs of the LFP. Otherwise, it's very hard to catch. So this, I hope, tells the usefulness of doing spike field analysis, which is very common in the field. Okay, and this brings me to the end of the LFP module. In our field, it's very common to do this kind of analysis, spike field coherence analysis. Any paper you pick up will have this sort of analysis because it very nicely captures the relationship between spikes and LFPs, which you can often not see if you only look at spike relationships, which is what people had done, in the past. All right, folks, we are done with LFP. And now we move on to EEG. There's some nuanced spike relationship analysis here, but we can skip all of that. Not really very relevant. Okay, any questions before I move to EEG?
I'm just doubtful that when we have the spike coherence, so using that, we don't know anything about the gamma and everything. So why do we then the data for spike relations is not reliable at all? Is
Yeah,
it
well,
like
it
that?
depends on how strong the coupling is. If the coupling between gamma and spikes is very strong, like this green case here, you would see that in the spike spectrum also. You will see that in autocorrelation also, and then it's all good. If it's not very strong, then it's hard to see. But if you did spike field coherence, you will always be able to pick it up. Okay, that's the difference. So people in the past used to just do spike spectrum or, cross correlation. You compute the autocorrelation or cross correlation and then compute the spectrum. And that works if the coupling is very strong. Otherwise, it doesn't work. Okay.
And so how everyone knows like so when people were working with spike spectrum only, do they have any means to find out whether the coupling is strong or weak?
Well, so the initial papers in the field were for conditions that induced very strong gamma. That's how people found out. People started looking at LFPs much later. So the earlier studies of Wolfsinger, for example, they were all spike, cross correlations and they even didn't look at the frequency domain. They're all based on cross correlations and the peak of the cross correlation function and so on. And there, we didn't look at phase differences. Spikes, tended to occur in synchrony. So that gave that idea to that binding by synchrony hypothesis. So notice here that spikes are occurring at a particular phase of the LFP near the trough, and the LFPs are all aligned to each other with zero phase lag, which means that gamma for one electrode and gamma for the other electrode are synchronous. So this logic suggests that the spikes are also synchronous to each other. Okay, so if I, this means LFP1 would be like this, LFP2 would be exactly like this in sync, and spikes would be occurring 2 milliseconds before the trough in both cases. So and the synchronicity can be captured in the cross correlation if it's strong enough. So earlier studies would just look at this as cross correlation. So this is as a function of time. They would look at the cross correlation function and the rhythmicity there. This LFP business came much later, and spectral analysis also came much later, and it allowed us to look at even weaker spike LFP spike interactions that couldn't be obtained using just the cross correlation analysis. Okay. So at the last part, our course on EEG analysis. So we have now finished 27 classes. I have not updated these three because I need to go to the lab to do this. I'm unable to go to the lab for at least another week. So for 21 days, the lectures are available on. So please download it. Don't rely on me. I don't know when I'll be able to go to the lab. So I'll locally save it, but I can't do this with my Mac. I need a PC to access this web page. So please download these three lectures, and then you'll have the full set. We will have classes on EEG. It's really not enough to cover this. I thought I would have more to do EEG analysis, but it didn't work out this way. We usually had a couple of more classes, but not that much more also. I think our pace hasn't been too. I think probably it's been too fast. It's okay that we have fewer classes. We will only cover these topics. In previous classes, we have also done PCA analysis, ICA analysis, and linear discriminant analysis. But anyway, those things are covered in other courses also, like what Arun and Rishi do, or it's usually gets covered in other courses. This is more signal processing heavy, which is sort of more aligned to what we have been doing. So in some ways, this is the important part that we are going to cover. So a couple of things before we get to the EEG. We'll only be able to start it today. So one thing which I think is very relevant for this particular group, you guys, because there are lots of biologists in this course this year. In previous years, we have had a healthy mix of engineering heavy students also, but this time, one reason we have gone a little slow here is also because many of you are seeing this for the first time. Okay, and in general, you often think that, hey, unless I understand everything in like really gory detail, I'm not able to make progress. And that actually, okay, there's two ways of looking at things. One is that you become like a super expert in what you're doing as far as analysis is concerned, and then you do an analysis heavy sort of approach. But there is also a part where you are just comfortable with the analysis and you are able to run it using some software that is given to you. But you're comfortable, but that's not the main research agenda for you. And this becomes true for many of you who are going to use these kind of analysis. But your main question will not be signal processing based, it will be a biological question. Like, for example, you record a monkey where you teach the monkey to do a particular task, or you are stimulating the monkey's brain with you're doing some micro stimulation, and then you are just using these techniques to generate the Fourier transform, and all you need to do is be comfortable with it. So just to give you an example, so when I'm running, let's say, plot rand n 1 comma 100, it generates 100 random samples that follow a normal distribution, Gaussian distribution. So if I did a hist here, say I did 1000 here, hist. It's a normal distribution with mean of 0 and 1. Now I am very comfortable doing this, and it's just a command for me. But if you now get down to the details of how a random number is generated, you would be stunned. It's like a research career of a person generating a random number which follows a particular distribution, and it's truly random, is very, very hard. Not a trivial question. So in some ways, I'm sort of not becoming an expert in random number generation, but I understand this code and I understand how to, display it, how to use it, and that's all for me. So in some ways, I'm using a software here. And a lot of the emphasis on this course is to make you guys sort of comfortable with signal processing. And it's meant even for people who are not going to be sort of experts and in signal processing and develop new tools and all that. You will just have to be comfortable, and it's an important stage for many of you to get in, because you won't be developing analysis methods. You will just be using the existing methods and develop the sort of the questions that are on the biology side. Okay, and your success as a scientist will depend on how you ask good questions on biology, not per se the signal processing. The assignment that I have given you, this assignment 3, and most of what we are going to do in the EEG part is actually geared that way. That is why I brought it up, right? We are going to actually think of this problem more from a software point of view, that how do I display certain things, how do I interpret certain things. And I'm going to use a particular software called Field Trip to convey some ideas. And what I want you guys to be is very comfortable with Field Trip, so that once you compute, collect your EEG data, and you know the codes and the concepts. Even if you are not super experts in it, it's okay. I mean, it's like you choose your battles, and if your battle is in the biology side, it's okay if you can just run it as a software. Okay, and a lot of emphasis in the course is to just get to you to that stage. So now is also, so before I start the EEG module, let me also just briefly mention what we are planning to do. So we have now 5 classes left. Okay, so we'll finish by mid-May, and what you have to do is start thinking about uh, um, grant proposal. So I believe now there are only 16 students. This time, there are fewer students than what we usually have. So you have to make teams of two. Okay, so choose a partner, project partner, and then you have to think of a proposal. The assignments that I have given you, they are such that if you spend enough time on it, you will sort of get all the answers. So the distinction between in grades, it is not going to be based on assignments that much. It's going to be based on your grant proposal. So I will send you details of how to write a grant proposal. I will also send you what people have done in the previous 2 years, and you have to really spend time thinking of a good question and how you can answer that question. And the good question will be on the back. In which case, you'll have to explain what the question is, and how you're going to answer, what has been asked, and what new you're going to bring. Or you have to think of a signal processing technique. This being a signal processing course, it would be preferable if you go signal processing heavy. But essentially, that's the main component of this part. This is this proposal now worth 35 marks. Please get to it sooner than later. You will have 2 weeks exclusively to work on this from mid-May to end of May. But start working on this soon. And as we look at this EEG data and start working on this, you will find it very useful to get used to field trip and see what others have done, and new sort of analysis methods that are not standard that have been incorporated, and then work on that. Okay. Okay, so with that, let me just start the EEG module. There's only a few minutes left, so won't cover much here. So EEG is obtained by sticking electrodes on the surface of the. You can. It's a very safe procedure. People who are meditating, infants, even prenatal babies, before they're born prematurely, you can record EEG from them. Completely non-invasive. Some part of metal that touches your head, and that's what you get. Okay, so usually there are different ways of putting the electrodes, some standard ways that by which you put these electrodes. So this, for example, is what is called a 10-20 system, and these electrodes are named based on which lobe they are on. So there's the occipital lobe, back of the brain, temporal lobes on the sides. This is the parietal area. This is the central area. This is the frontal area. This is FP1, I think is the frontal pole. Okay, so this is the 10-20 system in which these are the electrode positions. You also have denser grids in which there are more electrodes, and they come with fancier names than parieto-occipital, fronto-central, and so on. You can see here. You can just read this stuff. So it's kind of trivial. So I'm not going over too much detail. And these days you have very dense system also. In fact, we have a system like this in CNS, in Sridhar's lab. So 128 channel EGI. Okay, so it's already time. So let me not stretch it too much. I will start from the next class and tell you what we are going to do. Please download this EEG data from, it's there in the NS212 resource package. You will end up getting a resource, folders like this. There is some EEG data here, about 40 MBs, and some codes. And it's very intuitive. It's all based on this field trip package that you have to install. So please do all of that, and all our analysis will be built around this only. And new concepts will be fewer now. It's mostly going to be based on what we have already learned in the LFP module, and we'll sort of show it on the EEG data. And the new concepts that we are going to talk about will be in terms of source localization, which is the new part time. And choose your partner and send me an email, me and Krishna Kumar and Ankon, who are the two TAs. So there should be 8 emails now. Send me an email, say this is my partner, and maybe a week or so, you can send me what you plan to work on. I will send you guys details of what has been done in the past, what kind of a template I'm looking, and please start working on this while you're doing the assignment. It gels together because of doing an all in MATLAB. Okay, so that's that. There are any questions, I'll take them. Otherwise, we are done for the class. We'll start with the EEG module in the next class.